Welcome to Chapter 16, where we're going to discuss glycogen metabolism and gluconeogenesis. In this first lecture, we're going to start by discussing gluconeogenesis. And during the course of that, we'll discuss the overall pathway for gluconeogenesis, as well as the specific reactions involved in gluconeogenesis. And finally, we'll talk a little bit about how gluconeogenesis is regulated. In the next lecture for this chapter, we will talk a little bit more about glycogen metabolism. But for now, let's start by considering the overall metabolic processes for glucose. So glucose can be built up into glycogen. It can be broken down into pyruvate. And we discussed in glycolysis how glucose gets broken down into pyruvate. In this case, we're going to be discussing the opposite of that reaction, which is gluconeogenesis, taking pyruvate as a substrate and building it back up into glucose. So what is gluconeogenesis? Why is it happening? Where is it happening? Let's start thinking about that. So gluconeogenesis is the process of synthesizing glucose from non-carbohydrate precursors. This is usually pyruvate, but there are also other metabolites and intermediates that can be also fed into the gluconeogenesis pathway, some of which are converted to pyruvate or other intermediates. Gluconeogenesis occurs primarily in the liver as well as in the kidney. The liver is also a place where a lot of glycogen storage is occurring. And so it's important for the liver to be a sort of source of glucose. So if there's no more glycogen in the liver, that's one sign that the liver needs to start making more glucose, to sort of start replenishing those glucose stores so that they can be used as fuel for the rest of the body. And so the liver is making glucose via gluconeogenesis when it's necessary, and then using it as a fuel, exporting it out. However, gluconeogenesis is expensive. It has an overall cost of 6 ATP, and we'll go over where those ATPs come from in a moment. And so for this reason, or for a few reasons, it's not the exact opposite of glycolysis. While the starting point and the ending point are the same, or the opposites in each, right? Glycolysis, we start from glucose, end in pyruvate. Gluconeogenesis, we start with pyruvate, we end in glucose. Not all the reactions are exactly the opposite, and so the energy costs are different. In glycolysis, we had a net gain of 2 ATP, but in gluconeogenesis, we're going to have a net cost of 6 ATP. So first of all, gluconeogenesis is expensive. It's only done when necessary. And also, the other thing to keep in mind from this information is that not all of the reactions from glycolysis are exactly reversible into gluconeogenesis. The equilibrium reactions that we discussed during glycolysis are all the same, but the irreversible reactions from glycolysis are going to be different because they're not reversible, right? So that we can't just use the opposite of these because it's not, energetically favorable. So let's now take an overview and look at what's happening during gluconeogenesis. The entry point for gluconeogenesis is either pyruvate or oxaloacetate, which we see at the bottom of this figure here. Pyruvate down here, oxaloacetate over there in red. And lactate can be used as a source for pyruvate. It can be converted into pyruvate. And other intermediate metabolites from the citric acid cycle, as well as some amino acids, are able to be converted into oxaloacetate, which, as I just mentioned, can also be fed into gluconeogenesis when that's necessary. The first 2 steps of gluconeogenesis occur in the mitochondria, the ones highlighted in red here, but the remaining steps occur in the cytosol, just like glycolysis did. Fatty acid breakdown essentially can also be used as a fuel for gluconeogenesis because breaking down fatty acids is also a source of ATP, which can be used to fuel gluconeogenesis, which, like we just mentioned, has a net cost of 6 ATP. So let's look now a little bit more at the different enzymes that are used in gluconeogenesis as compared to the ones for glycolysis. So in this diagram, gluconeogenesis, those reactions, or the reactions that are different, are shown in red. The familiar enzymes that we saw from glycolysis are shown in blue. Like we mentioned before, the reversible reactions are the ones that are near equilibrium are reversible. Those are shared between glycolysis and gluconeogenesis. But there are 3 main steps from glycolysis that were irreversible that we discussed: the first step, the third step, and the 10th step. And those steps are when during gluconeogenesis, when we want to reverse them, we have to use different enzymes to make them spontaneous, right? So First, let's start with pyruvate and phosphoenolpyruvate, which was step 10 of glycolysis, turning PEP into pyruvate. For gluconeogenesis, to reverse this, to turn pyruvate back into phosphoenolpyruvate, there are actually two enzymes that are used. The first is pyruvate carboxylase, which turns pyruvate into oxaloacetate, and then the second step is turning oxaloacetate into phosphoenolpyruvate using an enzyme called PepCK. The third step of glycolysis, which we're seeing over here, was the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate using phosphofructokinase. To reverse that reaction, to turn fructose 1,6-bisphosphate back into fructose 6-phosphate, a different enzyme is used called fructose bisphosphatase, or fructose 1,6-bisphosphatase. And finally, the first reaction of glycolysis, which was the phosphorylation of glucose into glucose 6-phosphate, which used hexokinase, when it's reversed in gluconeogenesis, an enzyme called glucose 6-phosphatase is used to help remove that phosphate group. So let's talk about those reactions in more detail. So first we'll start with step 10 of glycolysis, which is the first step, last step of glycolysis, first step of gluconeogenesis. In glycolysis, as we discussed, shown on the top here, phosphoenolpyruvate is converted into pyruvate using the enzyme pyruvate kinase, yielding 1 ATP, or 2 ATPs for each half of glucose. For gluconeogenesis, two different enzymes are used. The first reaction is converting pyruvate into oxaloacetate, and this is using an enzyme called pyruvate carboxylase. And as the name might help you guess, the substrate for this enzyme is pyruvate, and it catalyzes adding a carboxyl group onto it, right? So we are adding a COO-, forming a carbon-carbon bond onto the end of pyruvate, and we form a molecule called oxaloacetate, which we'll see again in the citric acid cycle. Oxaloacetate then, through an enzyme called PepCK, or phosphoenolpyruvate carboxykinase, the K is for kinase. This is going to catalyze the addition of a phosphate group onto oxaloacetate, which is added over here to create phosphoenolpyruvate, which should be familiar from glycolysis. And so in this step, you can see each of these two steps, each uses a high energy phosphate bond, right? So we have an ATP being consumed by a pyruvate carboxylase and a GTP being consumed by PepCK. So again, this is energetically using energy, right? Instead of creating some ATP, we're using it. For step 3 of glycolysis, which was the phosphorylation of fructose 6-phosphate by PFK, or phosphofructokinase, to make fructose 1,6-bisphosphate, or F16BP, or FBP1, that reaction used energy by using an ATP. If we wanted to reverse that reaction, we would be creating an ATP, which in this case would be energetically unfavorable. So instead we use a different enzyme called fructose 1,6-bisphosphatase for glycolysis. And as the name might suggest, it's a phosphatase enzyme. So it straightforwardly hydrolyzes the phosphate group on the 1 carbon from fructose 1,6-bisphosphate to yield fructose 6-phosphate with an inorganic phosphate as the product, rather than making an ATP through substrate level phosphorylation. That's not what's happening. And finally, we see a similar reaction when we reverse step 1 of glycolysis. In glycolysis, this was converting glucose to glucose 6-phosphate, again by consuming an ATP using hexokinase to phosphorylate glucose on the 6 carbon. Again, reversing that would yield an ATP, which here would be energetically unfavorable. So in the reverse reaction, we again use a phosphatase enzyme, glucose 6-phosphatase. And this is going to, again, use hydrolysis of this phosphate group on the 6 carbon to yield glucose plus an inorganic phosphate. So let's now look more at the energetics of this. So we mentioned at the beginning of this lecture that gluconeogenesis is relatively costly. It uses 6 ATPs, and that's different from glycolysis, which actually yields 2 ATPs. So let's break that down a little bit. So in glycolysis, the first reaction using hexokinase and the third reaction using PFK, which we see here and here, are each consuming 1 ATP. And the reaction catalyzed by phosphoglycerate kinase from fructose 1,6-bisphosphate into PEP yields 2 ATPs. This reaction is a reversible reaction. That's why we didn't discuss it as a separate special step for gluconeogenesis, but it's reversed in gluconeogenesis. So while in glycolysis, the step is going to yield 2 ATPs, in gluconeogenesis, the step actually consumes 2 ATPs. The 10th step of glycolysis, using pyruvate kinase to convert PEP to pyruvate, also yields 2 ATPs. But in gluconeogenesis, there's 2 steps, right? Pyruvate carboxylase and PEP-CK. Each consumes an ATP, and in addition, there's the 2 halves of glucose that need to be made. So 2 times 2 halves of glucose makes a cost of 4 ATPs. And the other 2 gluconeogenesis reactions that we discussed that reverse glycolysis reactions 1 and 3, using fructose 1,6-bisphosphatase and glucose 6-phosphatase, have no net yield or usage of ATP. But ultimately, for glycolysis, if you add everything up, we use up 2 ATPs, we yield 4 ATPs, so there's a net gain of 2 ATPs that are produced by glycolysis. On the other hand, like we just mentioned, if you add up for gluconeogenesis, we're consuming a total of 6 ATPs. So ultimately, this whole process can be considered a process of substrate cycling, where you can go from glucose all the way down to pyruvate through a series of steps, and then all the way back up from pyruvate to glucose in another series of steps. This substrate cycling overall would cost energy, right? If you start from glucose, break it down, build it back up with 1 glucose molecule, glycolysis would yield 2 ATPs, and then you'd use up 6 ATPs to bring it back, ultimately with a net cost of 4. But ultimately, this is really important for your cells and in your body because it allows both pathways to be controlled in a coordinated way, and it also allows both pathways to be spontaneous, because otherwise they wouldn't happen. So, and it's really important to be able to control them separately, and having separate enzymes for these irreversible steps also helps to be able to control each of these pathways independently, so that one can be turned on while the other one is turned off. Because if you recall, for those equilibrium reactions that are reversible, if you speed up any of those reactions, you speed up the reaction in both directions. So those types of reactions near equilibrium would not be helpful for controlling one direction of this pathway versus another, which is why these irreversible steps that are highlighted here are particularly important for controlling which of these pathways is in use at any given time. So let's look a little bit more at that controlled regulation. So let's start with this substrate cycle with fructose 6-phosphate and fructose 1,6-bisphosphate. When we were discussing glycolysis, we talked about how phosphorufractokinase is the really important enzyme for regulating glycolysis. During gluconeogenesis, this step is also really important for regulation, but we're using a different enzyme here. We're using fructose 1,6-bisphosphatase to dephosphorylate fructose 1,6-bisphosphate back to fructose 6-phosphate. So when we discussed phosphorufractokinase, or PFK1, this enzyme was activated by ADP, AMP, which were signs of low energy levels, meaning that more glycolysis was necessary to make more energy. And it was inhibited by indications of high energy levels, which was a lot of ATP. PFK1 is also activated by a metabolite called FBP2, which is hormonally controlled, and we'll talk a little bit more about that in a moment. On the other hand, fructose 1,6-bisphosphatase, or FBPase1, is inhibited by FBP2. So you can already see here how FBP2 is a really important metabolite that can control this step in glycolysis and gluconeogenesis, because if there's a lot of FBP2, you activate glycolysis but inhibit gluconeogenesis. If there's very little FBP2, now we're activating gluconeogenesis but inhibiting glycolysis. So let's look a little bit more at how that works. Where does FBP2 come from? So it is primarily made in the liver, where we have a reaction, another substrate cycle seen here, with fructose 6-phosphate to create fructose 2,6-bisphosphate, or FBP2, and ATP is consumed by this liver PFK2, phosphorufractokinase 2, which adds a phosphate group to the 2-carbon of fructose 2,6-bisphosphate. which is shown here. And so that's why we call this FBP2. We can also call it F26BP, but it's mostly really important to differentiate FBP2 from FBP1. FBP1 is fructose 1,6 bisphosphate. FBP2 is fructose 2,6 bisphosphate. To remove that phosphate on the 2-carbon, that also occurs in the liver with FBPase2, which is a phosphoenzyme, and it's going to use hydrolysis of the phosphate group to help remove it and convert to make the product fructose 6-phosphate, or F6P. So PFK2 and FBPase2 are actually found on the same bifunctional enzyme. So when one piece of that is active, the piece that performs the FBPase2 function, there's ultimately going to be less F26BP or less FBP2. When PFK2 is more active, there's going to be more F26BP or more FBP2. FBP2. And so let's look at what actually helps to regulate the activity of those two aspects on that bifunctional enzyme. Like we mentioned, this is ultimately going to be hormonally controlled. So in the liver, when glucagon signaling comes through the bloodstream, this is going to activate the signaling pathway using adenylate cyclase, ultimately making cyclic AMP. Cyclic AMP is eventually going to activate protein kinase A, or PKA. And when we discussed our signaling pathways a couple of chapters ago, that's sort of where we stopped. We said PKA has several targets and sort of left it there. But here we're going to discuss some of the downstream targets of PKA that are involved in regulating PFK2 levels. So protein kinase A, as you can tell from the name, is a kinase. So it's going to convert phosphorylation of its substrates. And in this case, one of the substrates of PKA is our bifunctional enzyme, FBPase2 and PFK2. So when PKA is active, it phosphorylates FBPase2, which activates it, and ultimately helps to make less FBP2, because we're going to be dephosphorylating that 2-carbon. And this is ultimately going to help activate FBPase1, which, as we mentioned before, is regulated by the levels of FBP2. And this is eventually going to help activate gluconeogenesis. On the other hand, when we have insulin signaling from the bloodstream, this is going to activate through a receptor tyrosine kinase pathway, and eventually through a few steps is going to activate PIP3. PIP3 ultimately will activate PKB, and then PKB is going to eventually activate phosphoprotein phosphatase 1. And from its name, phosphatase, you already know that it's going to be dephosphorylating its substrates. So phosphoprotein phosphatase essentially has the opposite function of PKA here, where phosphoprotein phosphatase is going to dephosphorylate that bifunctional enzyme. And when it's dephosphorylated, the PFK2 portion is active. So when PFK2 is active, there is more FBP2 that is made, which is ultimately going to activate PFK1, which is going to help turn on glycolysis. So there's a lot going on here, but hopefully that sort of helps you see how hormonal signaling, as well as energy levels in the cell, are going to help to activate or inhibit each of the two enzymes here that are making and reducing levels of FBP2 that are ultimately controlling levels of glycolysis versus gluconeogenesis in opposite ways, so that when glycolysis is active, gluconeogenesis is inactive. When gluconeogenesis is active, glycolysis is inactive. And this helps to prevent that sort of unhelpful substrate cycle where you're just going to be losing ATP all the time. So with that, now you should be ready to answer the Chapter 16 Gluconeogenesis practice questions.